Boa tarde a todos, obrigado pela paciência. Uma vez mais estamos aqui para um CEBIMário mais. Quero agradecer então o Robert por ter aceitado participar desse seminário. Eu acho que vai ser bem legal a discussão que ele vai trazer um pouco sobre mudanças climáticas e assuntos associados a níveis trópicos. Então, thanks Robert for accepting this combiner for participating in this space. Very interesting your presentation or your talk today. Well, the seminar is yours in this moment. Thanks again.
Obrigado, Juan Pablo. Boa tarde. That's about the extent of my Portuguese. But thank you very much for having me. Like Juan Pablo said, my name is Robert, and I'm from Brown University currently, but most of the work that I'm going to be talking to you about today is between my work at Brown University as a PhD student and Woods Hole Oceanographic Institution, more recently as a postdoctoral scholar. Prior to Brown University, I was an undergraduate student at Oregon State University here in the United States. I actually did my master's degree at Universidad San Francisco de Quito, which is in Ecuador, and then followed on with my PhD at Brown University, where I did most of my research on fish communities in the Galapagos Islands. And I had the opportunity to work with Juan Pablo in Sergio Floater's lab in Florianopolis a couple of years back. So it's really nice to see you again, Juan Pablo, and to be here at least virtually back in Brazil. So I'll get right into the talk. And what I'm going to be talking to you guys today is about how my work has kind of unveiled some ways in which understanding the trophic ecology of fishes is really important in understanding how they're going to respond to ocean warming. Juan Pablo, you can see my screen. That's all working fine. I'll assume you are.
Yes, Robert.
Okay.
It's okay in this moment.
So I like to start my talks with this picture of a fish. This is one of my favorite species of fish, and personally, I think the aesthetics of fish ecology, fish biology, and evolution are all part of what drives me as a scientist. So this is Microperca olfax. This is a regional endemic grouper from the Galapagos. It's also found along the coast of eastern tropical Pacific countries. And the Microperca olfax has this xanthic coloration, which is kind of like an albino, where it's got this bright yellow pigmentation. They're really cool. I've only seen 3 of these individuals out of thousands of the species, so they're also quite rare. But apart from their attractiveness and aesthetic quality, fish communities can be a perfect model system for studying ecological change. They're the most diverse vertebrate group on Earth, with several thousand species described. They provide food and livelihoods for billions of people around the world, so they are obviously very important to humans. And because of their high diversity and their high functional diversity, and we'll get into what functional diversity means in a second, perhaps these fish communities can provide insight into the mechanisms of resilience and susceptibility to decline as we try to understand how different species are going to respond differently to the same anthropogenic pressures such as global warming. And one of the reasons that I'm studying fish ecology, and in particular the trophic ecology of fishes in the context of ocean warming, is the trend. This is from a paper by Sergio Floater from 2005. There's this global trend where we see a decreasing richness and abundance of herbivorous fish species as we go towards the poles and away from the equator. So that's, to put other words, as we go from warmer to colder oceans, we lose herbivorous fishes. So why is that? Well, that's several hypotheses have been put forward as to why that trend occurs, both in terms of abundance and richness of herbivorous fishes. And so here we're showing both latitude and temperature. So there's a negative relationship with latitude and a positive relationship with temperature. So that's produced this concept called the thermal constraint hypothesis, which is that at colder temperatures, ectothermic fishes cannot persist on algal diets. And there's a variety of physiological reasons why this might be true, but I won't get into that right now. And so if we assume that there are some, if we take as fact that there are some physiological limitations to metabolism and how fishes consume food and seek out food with temperature, then perhaps global assessments of how fish species might migrate with warming oceans need to take into account some of this trophic dependencies. And we can't just treat all fish species the same in terms of their response to warming. So this takes us to the Galapagos Islands, and this is where almost all of the work that I'm going to be showing you took place. So the Galapagos Islands are located about 1,000 kilometers off the coast of Ecuador, right on the equator in the tropical Eastern Pacific. The Galapagos are home to about 450 species of reef fishes and a pretty high rate of endemism at about 12%. So in addition to this, the reef fish fauna from the Galapagos is derived from a multitude of biogeographic provinces. There are species that have their evolutionary origins in the warm oligotrophic Indo-Pacific, which would perhaps be predisposed or pre-adapted to warm water oligotrophic conditions like we might be heading into with ocean warming. In contrast, species from the Peruvian province might be pre-adapted to colder and more productive waters, similar to that Humboldt current system that they evolved in, and might have a more negative response to warming waters. And then we have species that come from the Panamic province, which ranges between Ecuador and northern Mexico, and those species might be somewhat intermediate in terms of their proclivity to responding to warming oceans. In addition to those different biogeographic regions, there are a variety of fish traits that are represented in the Galapagos. So these are two papers, one by Stuart Smith et al. In 2013, and then another by our very own Juan Pablo and myself in 2019. And these studies have shown that relative to all other tropical reef fish faunas in the world, the Galapagos has A, very high functional diversity for the number of species that it has, and B, a high richness and biomass and functional dispersion of those species relative to other locations that are similar. And so all this is to say is that the fish fauna of the Galapagos Islands is unique and important both locally and globally. And so in addition to the location and susceptibility to perhaps phenomena such as El Nino, this is also an important location to be studying reef fish ecology. And in addition, this wide range of functional traits among species presents us with a potential map of ways in which we might be able to predict why species will respond in a particular way to ocean warming. For instance, trophic category or body size, or whether a fish lives on the benthos or in the open water, whether it's schooling or solitary. All of these different traits might have some predictive power in terms of how species respond to warming. So in that context, we had two major research questions that I've been exploring over the past 8 years. The first is, do species traits predict population responses to temperature variation in Galapagos reef fishes? And that's going to look at temporal change over time in response to El Nino phenomena. And then secondary, how do dietary requirements and food web structure vary with temperature? And so we're going to kind of hone in on one of these traits, which is trophic category, and look more in detail in terms of how trophic category will help us understand why some species respond in certain ways to El Nino events and why others don't. So in order to answer these questions, we've been sampling at 12 sites in the Central Archipelago. Since my PhD advisor, John Whitman, has been sampling there on the benthos since about 1999. I came in 2014 and added on top of that fish censuses. So again, we look at these 12 sites that cover an area more or less the size of the metropolitan area of Sao Paulo. And so you can imagine these are, it's a pretty long way to travel every day. It's about 60 kilometers from where we leave out of Porto Ayor and the Charles Darwin Research Station to get down to Gardner or up to Roca Cousins, for instance. So it'll take a couple of hours to get to some of these sites. So at each site, we undertake underwater visual censuses, 50 by 5 meter band transects, in which we count all fish, identify the species, and estimate size so we can calculate biomass. And we also use videos of reef fishes using GoPros that are placed remotely. So we place them via diver, and then we leave them to record remotely while we're doing our service intervals in order to get fish behavior in the absence of divers. We also at every site do CTD profiles, connectivity, temperature, and depth. These are 2 undergraduate students from our lab doing CTD profiles. And we also use in situ loggers, these little tidbit loggers that are recording temperatures at 2 different depths, 6 and 15 meters at each site every 10 minutes for the entire study period. So with that kind of temperature record, we can get something like this. So this is the temperatures between July 2014 and January 2019. And this is from one of our sites. This is Baltra. And we see a couple things pretty clearly here. One thing is we see some pretty strong seasonality. We see, repeated high warm seasons and cold seasons. But in addition to that, this was a particularly important period of ENSO phenomena. We had the pre-El Nino period, which was between late 2014 and early 2015. We thought that it was going to be an El Nino, but it then subsided. Then we had a full-on El Nino period. This was between the end of 2015 and early 2016. But then we actually had 3 successive pretty strong La Nina periods. So these were prolonged cold periods. Water temperatures were down around 16 degrees centigrade, which is very cold for such a tropical location. And the amount of food and upwelling that was being brought up to the surface by these La Nina periods created a huge boom in primary production, which we'll talk about later. So we have these, this temporal trend in temperature, and what we were doing was just tracking 12 discrete populations of the same species at each of these sites across all of these temperature fluctuations to look at that temporal change. So again, we looked at traits, these traits that describe each species, and looked at how each trait has the predictive capacity to tell us which species will respond differently to that ENSO variation. So these were the different traits that we examined. Those that have an asterisk came out as significant predictors in a linear mixed effects model. And the response variable for this was the ENSO abundance anomaly. So whether it was a positive or negative abundance anomaly during the El Nino, the peak of the El Nino relative to all other time periods over that 8-year sampling period. And in particular, diet came out with the highest partial R squared value of 0.18 in terms of predicting that ENSO response. So now I'm going to plot because we came up with this diet as being the most significant predictor in this model, we started to look more in depth at diet and how dietary category might help us understand how species are responding to temperature change. So here I've just separated out all of the species and aggregated them based on trophic categories. So we have higher carnivores, these are the top predators, herbivore detritivores, that are eating a lot of detritus and sand, herbivore algivores, which are mostly biting off pieces of foliose algae and filamentous algae, invertivores that eat mobile invertebrates like crabs and snails, invertivores that eat sessile invertebrates like barnacles, omnivores, which are kind of the other category, and then obligate planktivores, which are going to be very important. And then in the background, I've just shaded things based on the ONI, which is the Oceanic Nino Index, a measure of the strength of the El Nino and Southern Oscillation cycle. So warmer temperatures are indicated in red, colder temperatures are indicated in blue. And we've got mean biomass for each of these groups on the y-axis. And note that these are different y-axis scales. So first off, we see that there is some consistent downturns during the El Nino or directly after the El Nino period. We see this for 3 different groups in terms of statistical significance: the herbivore detritivores, the mobile invertivores, and the planktivores. In contrast, there are 2 groups, the higher carnivores and the planktivores, for which we saw an increase in abundance relative to all other time periods after these La Nina periods. And for the higher carnivores, it's a little harder to explain why this might be. We think it might be more because of migratory patterns for planktivore species like manta rays. But for these planktivores, we started to look much more in depth in that because these are actually reef fishes that are, that have to come back to the reef every night. And what we see is that this planktivore biomass is really reflecting that Oceanic Nino Index with about a 6 to 18 month lag. And that lag also varies whether you're talking about negative anomalies, which seem to be a little bit faster, versus positive anomalies, which might take a lot longer to occur because of the time that it takes for new fish larvae to recruit to the reef and grow to size to replenish that biomass metric that we're looking at. In addition to this, planktivores are very important throughout the community. They comprise about 36% of all reef fishes in the Galapagos. Here we're just looking at a rank abundance curve based off of this trophic category. And you can see that the 5 most abundant species are all obligate planktivores. 2 of the herbivores that are most abundant, that's Stegastes beebe and Prionurus laticlavius. It actually turns out are not really that herbivorous. We've been doing more and more work on diet using stable isotopes, and I'll get into that in a second. But it turns out that these are actually fairly plastic planktivorous herbivores, And so again, these are the most abundant species in the community. Not only this, we've been working on phylogenetic analyses for the reef fish community in the Galapagos, and we've described two divergences from non-planktivorous families towards planktivory just in the Galapagos. So one of them is colonus. This is a regional endemic, the Creole fish, which is a ceranid. These are large, groupers, generally piscivores, that diverge to become planktivorous. And then Xenichthyes and Xenoses are two genera that are also regional endemics that are grunts. Grunts tend to be invertebrores. These have diverged to become planktivorous just in the tropical Eastern Pacific. So this tells us that plankton and the ability to access that food resource really is fundamentally important for the fish community in the tropical Eastern Pacific and in the Galapagos in particular. So that was one of the main findings of my PhD research, and it led me to ask this question: well, how really do these dietary requirements change with temperature, and also how does the whole food web change with temperature, and how does reliance on one type of primary production such as plankton versus another such as algae or corals change when you have these temperature shifts? And so that led me to work at Woods Hole Oceanographic Institution. I've actually just finished a 1.5-year postdoc there with Simon Thorold. And Simon, with his former PhD student, Kelton McMahon, have been really leaders in the field of essential amino acid and compound-specific stable isotope analyses in general. And so what Simon and Kelton have developed is this methodology to track primary production from different end members. They're called different primary producers, such as, like I said, algae, phytoplankton, or corals. And you can actually track those sources of energy as they flow up through the food web in ways that prevent confusion by, such as a species changing its trophic category, its trophic intake with ontogeny, or if they're migratory. So we can actually sort of piece apart which are the causes of changes in stable isotope concentrations. So in this, again, came from our observations of changes during El Nino and La Nina cycles, and certain species being more susceptible and others being more resilient. But I couldn't go ahead and call up to whoever's making these decisions and ask for another strong El Nino event. It doesn't work that way, but we were able to take advantage of more of a spatial temperature gradient that also exists in the Galapagos once we kind of zoom out a little bit farther. So if you'll recall, our 12 study monitoring sites were right here in the central archipelago, but the Galapagos is much bigger than that, and there's different sea currents that hit the archipelago in different manners that cause a spatial gradient in temperature similar to that temporal gradient in temperature. And so here's a map of the Galapagos with the major currents. And in particular, we're seeing the upwelling of the equatorial undercurrent, or the Cromwell current, which is a deep cold water current that travels across the Pacific from west to east. And when it hits the Galapagos platform, it shoots up from about 3,000 meters deep to the surface, bringing this cold, nutrient-rich and oxygen-rich water to the surface, which really supports a huge proliferation of life. And it's one of the reasons why the Galapagos is known for being such a productive place. It's rare to see that much nutrient concentrations in the surface waters in a tropical location with constant tropical insolation from the sun. So basically, the closer you are to that center of equatorial undercurrent upwelling, the colder you are. And the farther you get from that, you start to deviate away from that source of cold upwelling water, and you start to get more typical tropical sort of stagnant surface ocean with warm temperatures around 26 degrees centigrade and lower productivity relative to that Western archipelago, which is 3 degrees centigrade on average, lower than in the Northeast. And a daily changes can be even more extreme. You can get down to about 12 degrees centigrade in the West and up to upwards of 30 degrees centigrade in the Northeast on any particular day. So expanding our study universe a little bit, we sampled from 3 kind of regions, the cold West, the central intermediate archipelago, and the warm Northeast. And at each of these sites, we collected fish and took tissue samples in order to look at how diets are changing across this gradient. We also did some sampling up in the two northernmost islands of Darwin and Wolf, but only observational. We weren't allowed to collect from those two islands because of their unique characteristics. So to look, to see what it's like underwater, Marchena Island, this is the northeast. This is very tropical. You see a lot of tropical fish. You see corals. Here's a Passalopora coral. They aren't true coral reefs, but there are sporadic corals throughout. In contrast, Fernandina Island is this subtropical cold water location with abundant macroalgae. It kind of looks similar to maybe Southern California or South Africa in terms of the underwater community. And you get these big algae feeders such as marine iguanas. In fact, you could probably just use marine iguanas as an indicator of sea state and productivity, because here in Fernandina, they're enormous, like 12 kilogram, the size of big dogs. Whereas in Marchena and Genovesa, they're tiny, less than a kilogram, and they're not even big enough to swim underwater and feed on the underwater algae like this individual is. So we did collections trips in 2019 that reflected the warm season, the 6 months of the warm season prior to that. And then again in January in 2020, which reflected the cold season. So we've got one of each replicate for these two warm and cold seasons. A ton of people were really instrumental in this work. Here's just a few of them. There's Anaïji Wróblewska-Łed, for those who know her. She also did her PhD in Sergio Floater's lab, and some colleagues from Australia who also came along. So for these collections, again, we're kind of going back to this question about this thermal constraint hypothesis. Is there evidence that at cold temperatures, ectothermic fishes can't persist on algal diets, or is there some sort of switch in terms of dependency with temperature? And again, we've got this condensed thermal gradient in which to test the predictions of the trophic constraint hypothesis, the thermal constraint hypothesis, with this diverse guild of herbivores. So here's some data. This is sea surface temperatures aggregated over the study period. You can see, again, we've got about a 3 degrees centigrade sea surface temperature gradient between the coldest sites in the west and the warmest sites in the north. These are all of our study sites. For chlorophyll, it was kind of a similar story. You get higher production in the west than in all other locations. However, it's not such a clear, there isn't so much of a gradient as there is here. It's really more either you're in these extremely high productive places or you're anywhere else. So we looked at temperature and chlorophyll A in addition to the biological predictors of the richness of herbivorous fishes and the abundance of herbivorous fishes in terms of trying to generate some predictions about what would drive differences in herbivory rates. So again, I'm just going to kind of dive right into some of the data. These are all of the species that we observed feeding on the benthos, 27 of them, I think. And then we have several different families. A lot of these are the typical tropical herbivorous fish families: Acanthuridae, Labridae, Pomacanthidae, Pomacentridae. Some species were only observed in the warm oligotrophic north, such as Acanthurus nigricans and Forcipiger flavissimus. These are well-known tropical species. In contrast, there were also some herbivorous fishes that were only seen in the colder west, such as Nextalosis latifrons. This is a fairly cold water herbivore from the coast of Peru and Ecuador. So it makes sense that this would perhaps be more associated with cold water regions, but it's still feeding, so it's indicating that there isn't this strict limitation of algivory in the 23 degrees centigrade range. So looking at these predictors against herbivory rates, we've got each of these now. So there's abundance, chlorophyll, SST, and richness. 3 of those predictors had significant relationships with herbivory. They were also tightly collinear, so it was difficult to really partition which of those 3 was most important. Chlorophyll was mostly dominated. The trend was dominated by these 2 sites. Again, those are those 2 extremely high productive sites from the west side of the archipelago, so there wasn't much of a relationship there. But we wanted to dig into this a little bit further because there were those collinear effects. So we actually looked at the relationship between sea surface temperature and herbivory after accounting for those richness and abundance effects with temperature. And what we found was that after accounting for abundance, there was no relationship. So there's no relationship between bites per capita and sea surface temperature, which would suggest that it's more of there's a greater abundance per species with warmer temperatures, or they could be spending more of their time throughout the day feeding, perhaps because of a higher metabolic need for intake because of higher water temperatures. But certainly the richness effect alone is not enough to explain this, as you can see there. Also, when we dig into individual species, the trends are not consistent across all herbivores. Holocanthus passer, that's an angelfish, and Prionurus laticlavius, a surgeonfish, they both had this predicted trend of increasing herbivory rates with temperature. And yet, Scarus gobon, which is another important herbivore on the reef, while it initially had this increasing relationship with temperature, the last two sites, which are Darwin and Wolf, those farther north sites, had lower herbivory rates, and that actually could be an effect of the island area. Those are smaller islands, and these are territorial and harem-forming fishes, so they could actually be limited in terms of area of reef available. Also, there could be dispersal limitation because those islands are actually not in the path of currents coming from other Galapagos Islands. So it could be a lack of dispersal and larval settlement to those sites. Then for Stegastes bb, we have an actually negative relationship between herbivory and temperature. And so this could suggest that A, the species is not accessing algae, or it's accessing more plankton, or B, that the greater abundances of this species in colder sites is simply generating these higher herbivory rates at colder sites. So with the collections, we were also able to look at tissues and look at stable isotopes. So I'm going to have to kind of blow through this a little bit quickly, but again, what we're doing is we're looking at just essential amino acids. These are amino acids that do not fractionate as they go up through the food web. So you're getting a very faithful representation of the primary producer sources that generated the energy that is making up the biomass of this individual. We can actually say that, 60% of this shark or whatever fish we're looking at is generated from algae versus coral versus plankton, or at least that's where we're hoping to get to. These are some folks that have been working with me out at Woods Hole Oceanographic Institution. We're only just now starting to generate some data because of delays because of coronavirus, but I'll show you a little bit about what we're finding out so far. So this is a principal coordinates analysis where we've taken 5 essential amino acids and we've condensed them into just 2 axes, which explain over 90% of the variation in the model. And then we're going to map all of our primary producers and all of our fish onto that multivariate ordination. And there's also, you can see where the different amino acids kind of fall out. So first of all, we can see that this method does separate and segregate out our distinct end members, or our primary producers. I'm going to reorient the axes a little bit just to center them, and then add fish on top. So now we're looking at primary producers in black, planktivores or supposed planktivores in orange, herbivores in purple, and generalists in green. So we see some pretty good overlap between what we expect to be planktivores and plankton, a little bit less in terms of herbivores to algae, and the generalists kind of fall out all over the place, and that's because they're generalists. But digging in a little bit more in terms of these species, we see that just looking at planktivores now, so these are 4 different species of planktivore, they are not necessarily overlapping. They're eating perhaps different components of the plankton with different phytoplankton sources. Even within a species, we found that locations that have high upwelling versus low upwelling also differ in diets, and we're still kind of trying to understand why that might be. But looking at the case of this yellow-tailed surgeonfish, Prionurus laticlavius, we start to understand some of this dietary plasticity that I was hinting at before. So again, we're looking at the plankton, algae, and detritus and members. And in blue are samples for Prionurus laticlavius that we're taking during the cold season. And in orange and red are the samples that we're taking during the warm season. And then we've got, based on the location of upwelling, we've got high upwelling in squares, low upwelling in circles, and intermediate upwelling in triangles. And there's 2 main takeaways from this slide. The first is that the differences between upwelling regions, so that's looking at the distance between red points and the distance between blue points. The distance between red points, so that's the high upwelling location is greater than that's the low upwelling location, is greater than the difference between the low upwelling region. So that's to say that diets are changing between seasons in the low upwelling location more than they are changing between seasons in the high upwelling season. At the same time, the differences between seasons is greater in the low upwelling region. So these two data streams are, what these two sort of conclusions are telling us is that fish that live in the oligotrophic Northeast have more variable diets between seasons, and the differences between high and low upwelling regions are maximized during the warm season, which is when that upwelling of cold nutrient-rich water is relaxed, but there's probably a minimal amount of that delivery coming up to the surface waters always in the western side of the archipelago, but the tendrils of that current that wrap around the north side of the island to reach those northeast islands aren't making it during the warm season, and that's causing those fishes that live in the warm oligotrophic north to have to switch to an alternative, less desirable source of food, perhaps algae or detritus. And while we thought that this sort of plasticity would confer some resilience to El Nino, it doesn't really look like that. Rather, we think that the degree to which you rely on plankton predicts the degree to which you decline during El Nino. That's what we think it's going to come out as. So to conclude, really quickly, we found that planktivore abundance and biomass are both negatively correlated with temperature, whether we're looking at ENSO in this temporal pattern or the spatial gradient. And that herbivore abundance and richness are positively correlated with temperature. In terms of food web structure, we found that herbivory rates are positively correlated with temperature, supporting the temperature constraint hypothesis. Also, that there is some dietary plasticity in these herbivore generalists that also might support this thermal constraint hypothesis. And finally, we found that the effects of temporal variation in upwelling due to seasonality mirror spatial differences in generalist diets. And so perhaps that space for time proxy using the spatial gradient of temperatures in the Galapagos is a valid proxy or representation for what goes on during these El Nino periods. With that, I'll say I'm going to be starting at University of Florida in January of next year, and I would love to collaborate and talk with you folks more about how we could, work together and answer some of these questions going forward. And with that, I'd like to thank Juan Pablo for inviting me and all of you for being here, my funders, field assistants, lab assistants, and all those that have helped to make this work possible. Thank you.
Thanks, Robert, for the presentation. Very, very nice. Know more about your work in this moment in Galapagos. The last year I following your works, and it's very interesting.
Thank you.
See this advance. Ok. Pessoal, agora eu vou, como escrevi aí no chat, a gente tem um espaço para perguntas, temos ainda uns 20 e tantos minutos para a gente conversar ainda mais com o Robert aqui sobre o trabalho que ele apresentou hoje. Well, Robert, I will wait for 1 minute. Different question about the people here assisting in this moment your talk. But I have several questions with you about for example, the use of the different traits.
Yeah.
You showed in this moment, for example, difference in the tropical species and subtropical species, and you see the difference in the biomass and for different species. But my question is, you try to see another traits, for example, both sides, you see changes in the both sides for different species in different place or not?
I'm sorry, the last bit I didn't quite catch the question.
Yeah, okay, more easy. You see difference in the both sides and the biomass in the time?
So between regions? Yeah, that's a really great question. So we see for most species larger body size in the colder, higher productive locations, so in the western side of the archipelago, and that's a pretty common phenomenon. Organisms that live in colder temperatures tend to grow to larger size. We don't know if they're growing slower to a larger size and growing older, like they're surviving longer, or if they're actually growing faster. But we've taken otoliths from all these fish, and we'd like to actually look at like growth increments and get a sense of how old these fish are and whether that's kind of helping explain this. But we think it's probably some combination of growing slow and long in low temperatures and having a greater abundance of the more energy-rich plankton food in the West.
Perfect. Well, I tried to read the question here, a comment for the other people, and after I tried to new question with you. Well, Sergio Flotter is writing here, Scarus Goban, Darwin and Wolf is more exposed and turbulent weather, but is not good for herbivores.
Yeah. Well, of course. Thanks, Sergio, for pointing that out. In fact, we've had reviews come back from this paper with Kendall Clements as a reviewer, so you can imagine he was pretty harsh on that. But yeah, he said the same. You know, how can you treat this species as an herbivore when they are accessing a whole bunch of different resources? And what we're hoping to do is use the stable isotope information to validate the herbivory information and get a sense of what they're assimilating and not instead of just what they're feeding on. We also reduced, you know, we constrained our analyses to those species from global databases developed by David Muilo to categorized as herbivores, so that we basically use that global database as our information to say this is an herbivore or not, because dietary information is lacking for Galapagos. So that was that first step. You know, we were using that type of categorization from a global study to label our species in the Galapagos in the absence of dietary information from the species. Now that we're producing our own dietary information for these species, we don't have to categorize each fish as being a planktivore or not because it was labeled as such from a global study, but we can actually use our data from the Galapagos and answer that question.
Well, you know, this moment you have one data paper is available for different trends. It's published this year in ecology, and you have the more specific trends. Maybe you can use it for this. Yeah. Another suggestion about this point, maybe you can use the trophy level and don't use the specific, because it is one continuous variable, and you can see the difference between the tropical and subtropical seed is very different when you try to use only the diet, for example, in this case.
That's a very good suggestion. Thank you.
Well, you have another question here for the Carlos Eduardo Ferreira. He's split. Hi, Ralph. Nice talk. Happy to use results. But can tell more about you considering detritus for amino acids analysis?
Yeah. So are you talking about? Sorry, can you just ask the last bit of the question about the amino acid analyses?
Yeah, Oscar, I can repeat.
The last bit.
Yeah. Tell more about what you consider detritus for amino acid analysis. How do you consider detritus in your analysis?
Sorry, that word.
Yeah, you can see the comment for the Kadu is the chat.
I'm not able to see the chats myself for some reason, but I can talk a little bit more about the stable isotope analyses. So traditional stable isotope analyses use bulk tissue stable isotopes. And the reason that we didn't opt for that is that there are kind of three reasons why the fraction of heavy carbon, so this is carbon with an extra neutron, carbon 13, there are three reasons why heavy carbon might change in a fish from one point to the next, or from one individual to the next. The first, which is what we are interested in, is the source of energy and the primary producer that actually takes up the carbon into their tissues from the available carbon in the environment. Each primary producer has its own mechanism for doing that. And based on divisions, evolutionary divisions, the farther back you go in terms of those divisions, the more different those assimilation methods and metabolic pathways are. And so if we delve back far enough and we compare an algae, like a red algae, a rhodophyte, from a zooxanthellae and a coral, the division between those primary producers is very ancient, and they are they're taking up heavy carbon and assimilating it into their tissues at different rates. So that is what we're most interested in. But if we only use bulk stable isotopes, if a fish ate a coral and has a physiological process that assimilates proteins into its body that's different from a different fish that also ate the coral, you might get different measures, even though they both ate the same thing. In addition, fish can feed in one place and then migrate to another place and feed on the same thing. But if the coral in one location is bathed in water that has a different isotopic ratio than corals at the first island, then again, you could have differences even though they're eating the same thing. So those are the problems with bulk stable isotopes, that they could be fractionated as they move up through the food web due to trophic level, due to location, or because they're using different primary producers. And that third one is the only one that we're interested in. And so that's why we use amino acid specific stable isotopes only for those amino acids that are essential. They are not created by the animal, they are only created by the primary producer, and they don't fractionate as they go up through the food web. So hopefully that answered some of your question, Kailu. I'm sorry, but that's why we're using this technique. I'm also interested in using bulk stable isotopes to look at trophic position, where species are relative to each other. Because eventually what I would like to do is integrate across the whole food web from the top predators to the bottom, how much of this food web relies on plankton, how much of the food web relies on algae, how much of the food web relies on coral, and how much on detrital microbial processes, and how that varies spatially and temporally. Eventually, I would like to come to that conclusion, because then you could actually use El Nino cycles and the expected change in delivery of nutrients to the surface as sort of whole ecosystem measures or predictors of productivity, which could then have implications for fisheries yields, etc. So that's kind of the place we'd like to go with this.
Okay, I think is the Kadu complete the question is where the detritus come from.
Oh, where does the detritus come from? So there are several microbes in the ocean that are able to capture carbon via non-photosynthetic methods like chemosynthesis. And so in the detrital pathway, there is injection of new heavy carbon into the food web that isn't accounted for by other primary producers. So and we use, as a proxy, we use sea cucumber tissue because they're detritivores. So that's where we get that sample from.
Okay. You have another question here for Sergio. What do you think is more important pathways for energy in Galapagos, both out for algae or coming from plankton?
Coming from what?
For plankton.
I think it's so. I've talked with a couple of people that have been doing similar work in different ecosystems, even in like tropical coral reefs, and it's actually coming to our attention that there's a greater reliance on planktonic subsidies than we earlier believed, even in places that are supposedly oligotrophic. So in the Galapagos, where it's extremely productive in a high plankton resource, I would expect it to be like 90% reliant on plankton. I don't know, putting a ballpark figure out there, but even these species that are supposedly herbivorous are eating plankton. I've got videos from these salp blooms. We get these big blooms of colonial salps that are in rings like this, about this size, and everybody eats them. I mean, absolutely everybody. There are blennies and gobies coming out of their holes to eat them. There are pelagic fish coming to the reef to eat them. There are herbivores leaving their territories to eat them. So I think it's a matter of availability. If there is this nutrient-rich food that's right there and accessible, even if your body isn't built for it, there is this adaptation to take advantage of it, and it's available so often in the Galapagos that it's driving a lot of what's happening.
Okay, I have another question about your presentation. Galapagos is a very interesting place because you have the transition zone, because you have the hot water and cold waters is put together. But my question is, do you see it in the last years tropicalization arriving for the tropical species more in the subtropical size, or the situation is very stable, because when you comparing, for example, another wars in Australia, you usually do observe more tropical species
Yeah.
in your
Yeah.
geographical.
Yeah, that's a very good question, and certainly larval supply and the arrival of propagules in Galapagos is a big black box still. We don't know much about internal dispersal between islands or the arrival of propagules from elsewhere. During the El Nino of 2016, we saw tropically derived species. I remember in one particular, like a red-shouldered wrasse, I think, which had only been observed in Darwin and Wolf. Prior to that, we saw it at Baltra, which is the central intermediate sort of archipelago. I didn't see like, tons of like tropical angelfish and butterfly fish all of a sudden everywhere. It wasn't like that. But there are anecdotal reports of like lionfish arriving in the northern archipelago during El Nino periods, like 97, 98. El Nino, I think there was a report of a lionfish. So that probably was brought across the Indo-Pacific. I don't know where else it could come from. So they definitely do arrive. But, at the heart of the matter, I think for us, what we're trying to understand is, are the predictions and sort of expectations of fish in particular changing their ranges? A lot of these predictions come from experiments where you put a fish in a tank and you warm it up until the fish dies, or you look at previous or current day associations of fish with different thermal, isotherms. And so those types of analyses and experiments are really informative. But at the end of the day, we think that, and there's more evidence growing, that there are going to be limiting resources that get in the way, that prevent fish from surviving up until they reach a true thermal threshold, such as food or biogenic habitat. If you're a coral specialist and it becomes too cold for corals, or because of warming, there's too many bleaching events and there's no coral, even though you could theoretically survive those temperatures, the necessary habitat for existence doesn't exist. And in this case, in Galapagos, it's more about food, I think, than habitat, because there's so much available reef structure because of the rocky reefs that we have. But getting at these traits that sort of mediate responses to warming and help us predict which species will prevail or not, I think that's kind of where we need to go.
Ok. Thanks, Robert. Pessoal, mais perguntas para o Robert antes de a gente finalizar. Ainda temos tempo para mais perguntas a mais. Tem uma pergunta aqui. Do you analyze migratory species during observation or only local fauna?
Yeah, no, we excluded migratory species such as mantas, just because that seasonal effect could be so strong, irregardless of what they're eating. We only worked on resident reef associated fishes.
Okay. Mm-hmm. Well, you know, Galapagos, I think, is the only island you lost in one endemic species.
Yeah.
Very records is.
So the last I saw, it's still undetermined in the IUCN list in terms of whether it's extinct or not. But what Juan Pablo is referring to is Azarina eupalama, which was a endemic damselfish from Galapagos that hasn't been seen since I think it's the 82, 83 El Nino. So as a result, this was a damselfish that was believed to be almost planktivorous, if not partially planktivorous. And so the loss of planktonic food during El Nino could have driven that species to extinction. We also detected an ulcerative skin fish disease during the 2016 El Nino. It hadn't been described before, but it's possible that it was around, and just because there weren't as many divers in the water, it wasn't detected. But that disease could have also contributed to the downfall of that species. But yeah, like Juan Pablo said, that is, as far as we know, the only fully marine fish to go extinct as a result of climate change. And that could be a canary in the coal mine in terms of, there could be a lot more species that have that same fate in the future.
Yeah, but my question is more specific. You see it in the the biomass or abundance decrease in different important endemic species, for example, or do you see the...
Yeah, so Zenosis jessii, I think I mentioned. I'm just going to run back to that. Yeah, I don't have the population trends for that species right now on my computer, but this is Zenosis jessii. It's an endemic planktivore. This species was not seen at all during El Nino, and probably about a year after that. But during the La Nina period that followed, there was this huge recruitment event. I mean, there were clouds of these little 1 to 2 centimeter larvae just covering the rock. You almost couldn't count other fish because of them. So I think that these are just boom and bust species. You know, they they have really, really good times during La Nina, and the few that survive in El Nino period carry on to replenish the population. So there might be, like, you know, repeated bottlenecks and expansions of those populations. And this is, again, a local endemic to Galapagos.
Mm-hmm. Well, Sergio put another question here: is the gas to be more okay con temperature?
More what?
Is okay with temperature?
Well, it was one of the species that had the highest prevalence of skin disease during the skin disease outbreak. It has lower abundances in the warmest locations in the Galapagos. So, you know, there is negative correlation there. Does it do okay? I mean, it survives. You know, there's plenty of Stegastes bibi in the warmest northernmost islands, but they're smaller and they're less abundant than they are elsewhere. So, you know, theoretically, at some point, it will reach a warm limit. Currently, you know, they're one of the most abundant species in the Galapagos, so I think they're doing fine. But it's a good question. You know, if they have that negative relationship, at some point it might come to a head.
Okay. Yeah, this is a very interesting question because, for example, here in Brazil, I try to use the temporal variation for the endemic species, and I have different Pomacentridae, and I see one decreasing, for example, in tropical species in Fernando de Noronha, Atol das Rocas, for example.
Mm-hmm.
It's not, sorry, one climatic change explain this tendency, but it's important checking this part.
Yeah, certainly. Whenever you're working with endemic species, it adds, you know, a whole layer of complexity and interest. Yeah, you know, I think that these phylogenetic divergences in the tropics and Pacific are super interesting. It's obvious that there is this important food resource that's being accessed to the point of even like diverging away from your normal life history strategy. So, yeah, these endemics and the way that they adapt locally to those local conditions is really fantastic. Yeah, it's a lot of cool stuff to study.
Okay. Well, thanks again, Robert, for your talk today. Very interesting.
Thanks so much for inviting me. This was a pleasure, and I hope to make it back to Brazil soon. And I hope that all of you guys are doing all right with the pandemic and all the challenges that it's presented.
Okay. Pessoal, então, muito obrigado a todos que estão ainda nos assistindo. Então, eu quero já aproveitar para convidar vocês para a nossa próxima palestra, a próxima quinta-feira, onde vamos falar um pouco sobre lixo em Fernando de Noronha. E vejo vocês até a próxima quinta-feira. Oh, desculpa. You have one new question here. This is for the new, sorry. It's the last question here. Is there fixed time? I don't know. It's specific. Read this question here. You can sit, Robert. Hello.
I can't see the chats for some reason.
Yeah, stop for sharing the screen. Stop for sharing.
Stop sharing?
Yeah, and you have the assets for the comments. The right side.
Yeah, I see the chat window, but nothing.
Yeah, you have.
Okay. Is there a fixed time lag between La Nina and La Nina events? Okay. Yeah, so it's not fixed. It's generally about a 2 to 3 year cycle between the warm and the cold ENSO, or El Nino phase, but then there's also the Pacific decadal oscillation. So about every 10 or 20 years, you get an extreme event in one direction or the other. So there is somewhat of a fixed, it's a regular interval, but it's not perfectly fixed, let's say. And then which species does increase? That's a great question. So there were a couple of species that we saw increasing following El Nino. Trying to remember like the specifics of them. There wasn't any sort of unifying characteristic, though, if that's what you're asking. It wasn't like all of the herbivores or all of the piscivores or all of the tropically derived species. We really thought that biogeographic center of origin was going to be an important predictor, but at the end, it really didn't fall out. And so it might be that it's much more nuanced. For instance, there could be interactions between the factors. If you're from the Peruvian province, but you're not a planktivore, you're fine, something like that. But it starts to get to be so high dimensional that it's hard to really come up with like a complete sort of answer to that question. Sorry.
Okay. Well, thanks again, Robert. Obrigado a todos, pessoal, e vejo vocês na próxima quinta-feira. Até a próxima.
Obrigado.